Hello, today we're going to be looking at human immunity. This presentation will be looking at non-specific defences against diseases, specific immunity and the biology of vaccination. This first section of the presentation intends to explore non-specific defences against diseases. So firstly, what is an infection? An infection occurs when microorganisms invade the body, typically through the mouth or nose or through cuts in the skin. These microorganisms are able to increase in number and cause reactions in the infected, such as fever, inflammation, and pain. Different microorganisms cause different types of reactions. For example, bacteria, which are single-cell organisms and can survive in or outside of the body, cause illnesses such as meningitis and strep throat. Viruses, which require a host to replicate and survive and cause diseases such as a common cold and chickenpox. Fungi, which causes skin conditions like athlete's foot and thrush, and parasites, which cause parasitic conditions like scabies. So how are these infections transmitted? There are four methods of transmission. Firstly, direct contact, which includes person to person through touching, kissing or sneezing, for example. From animal to person, such as when bitten by an infectious animal, or from mother to child, as if a woman has an infection, it can be passed through the umbilical cord to the child. Indirect contact occurs through contact with contaminated surfaces or objects or through the air. For instance, if an infected person touches a doorknob, the germs will remain on the doorknob and the next person who touches it can also become infested. On the other hand, airborne transmission occurs when an infectious disease travels through the air, such as with sneezing. If somebody sneezes without covering their mouth, the germs can travel up to 8 meters through the air. Infectious diseases can also be spread via insect bites by vectors or carriers. This is how ticks and other insects like mosquitoes typically spread. Additionally, food can be contaminated with E. coli bacteria, for example, which causes food poisoning and can make a person feel extremely ill. Now we have a better understanding of what infections are and how they're transmitted, we'll look into how our bodies defend us from these diseases. There are two non-specific or innate defences which function to prevent pathogens from entering the body and to get rid of invaders. These defences are automatically triggered by genetic programming. Firstly, we'll look at physical barriers. The largest organ in the body and our most notable physical defence is the skin. The skin's epidermis acts as a waterproof barrier, preventing toxins, irritants or microorganisms from penetrating. If the skin is cut or grazed, it will quickly form a scab that will serve as a temporary barrier while the skin underneath heals. The other physical barriers, mucous membranes or mucosa, that lines the immediate internal system, such as the eyes, ears, throat, as well as the gastrointestinal, respiratory and vaginal tracts. These membranes trap pathogens by producing mucus, and the human body actually has more than 200 times more mucosa than skin, making it the largest protective barrier. In addition to physical barriers, there are chemical barriers which shield the body from infection by eradicating microorganisms on the exterior of the skin, on the exterior of the body, at body openings and on the interior of the body's linings. Saliva, tears and perspirations all act as carrier barriers. Lysosome, which is found in all three, acts as a vital antimicrobial agent to destroy pathogens. Additionally, a very thin, slightly acidic film is created on the skin surface by the acids secreted by the sebaceous glands in the dermis of the skin in order to prevent germs, viruses and other harmful pollutants from penetrating. Several infections are unable to survive the acidity of urine and vaginal secretions, and also most pathogens that enter the gastrointestinal tract through food or water are eliminated in the stomach via stomach acid and digestive enzymes. So the second line of defence consists of a range of non-specific responses to a pathogen that enters the body. First, we have inflammation, which is the natural response of the body to infection, injury or tissue damage. When a pathogen enters the body, inflammation is triggered and white blood cells are sent to the site of the infection to fight off the pathogen. Secondly is fever, which is another way the body fights off infection. A fever is an increase in the body temperature which helps to kill off pathogens, as many cannot survive at the higher temperatures. Phagocytes are a type of white blood cell that engulfs and destroys pathogens. These cells include macrophages and neutrophils. Further helping in the breakdown and removal of pathogens from the body are complement proteins, a collection of 30 small blood proteins, and lastly are cytokines, which are small proteins that inhibit the reproduction of viruses, as well as having other functions such as signalling to the immune cells how to fight threats and repair injuries. The inflammatory response is one of our second lines of innate defence. It works to isolate the area, prevent pathogens from spreading and speed up healing. An inflammatory response involves a number of cellular activities when an injury occurs. The physical result is the dilation of blood vessels, which creates openings in the cell wall, enabling large blood cells to pass through, boost blood flow and cause inflammation. Due to the excess blood and proteins drawn to the infection, the area may appear swollen, feel warm to the touch and frequently hurt. We can see an illustration of a wounded skin area in this diagram. Chemical signals from the cells have been alerting phagocytes, more specifically macrophages, to move into the area and absorb the bacteria into a vesicle known as a phagosome. This process is called phagocytosis. The phagosome fuses with lysosome, a vesicle containing digestive enzymes designed to disintegrate the phagosome's content. This fusion is referred to as phagolysosome. Macrophages formed from the monocytes are particularly important as they recognise, engulf and destroy target cells. This next section will cover specific immunity. First, we'll look at the third line of defence, which is also known as acquired or adaptive immunity. It's a more specific and targeted response to pathogens in the innate immune system. Acquired immunity involves the production of immune cells and molecules specific to the pathogen, known as antigens, which trigger the response. Once the immune system has been exposed to a pathogen, it can remember the pathogen and produce a more rapid and effective response if exposed to it again. Leukocytes, also known as white blood cells, are essential components of the immune system and are cells that help the body fight off illnesses and external invaders. White blood cells are divided into 5 subtypes that are produced in the bone marrow. These include, but not limited to, neutrophils, which are the most prevalent type of white blood cell. They function by attaching to the walls of blood vessels in an attempt to prevent germs from entering the bloodstream, and are also first cells to detect an infection. Monocytes, which are the largest white blood cell but less common than neutrophils, they develop into macrophages when they exit the bloodstream to perform the role of phagocytosis. And lymphocytes, which are small round cells important to the humoral system. B cells, which account for 10 to 15% of all lymphocytes, are responsible for producing antibodies, which are proteins that recognise and bind to specific antigens. These antibodies can neutralise the pathogen, mark it for destruction by other immune cells, or prevent it from infecting cells in the body. The presentation of antigens by antigen-presenting cells, or APCs, activates B cells, causing them to produce the antibodies specific to the antigen. T cells, on the other hand, are responsible for killing infected cells and insistent in the activation of B cells. There are 3 main types of T cells: cytotoxic T cells, helper T cells, and suppressor T cells. The body uses cytotoxic T cells to fight off infections, reject tissue transplant, and generate an immune response to some tumours. When trying to kill an infected cell, a cytotoxic T cell binds to it and releases perforin right into the cell. Helper T cells are the most common type of T cell. They release lymphokines that help control the other parts of the immune system by stimulating other types of T cells to grow and attack. They also help B cells to grow and mature into their active form. Finally, suppressor T cells can inhibit the function of helper and cytotoxic T cells in order to regulate other immune system cells, preventing them from causing excessive damage to the body's own tissues. Immunoglobulins, also known as antibodies, are produced by B cells and they bind to specific antigens. There are 5 types of immunoglobulins: A, D, E, G, and M. Each type has a different role in the immune response. For example, IgM is the first antibody produced in response to an infection, while IgG is the most abundant antibody in the body and can cross the placenta to provide immunity to a developing fetus, while their humoral response is ineffective. Self-tolerance is simply the body's ability to distinguish between itself and non-self tissues, which is crucial in preventing an immune response against its own tissues causing autoimmune diseases. The immune system achieves this through a mechanism of antigens. The 2 fundamental factors which allow the immune system to tell the difference between itself and foreign bodies are 2 sets of cell surface proteins known as major histocompatibility complex, MHC1 and MHC2. MHC1 molecules are on the surface of all nucleated cells and show the immune system what's happening inside of the cell. They present small pieces of proteins from inside the cell to CD8 plus T cells on the cell surface. If the T cell doesn't recognise the antigen, they will attack and destroy the infected cell. MHC2 molecules, on the other hand, are only present on antigen-presenting cells. They present small pieces of protein from outside the cell to CD4 plus T cells. If they don't recognise the antigen, they will activate and destroy the pathogen. The human immune system, however, does not respond consistently to foreign antigens. This means the degree of response varies depending on how similar the antigen is to self. Failure of self-tolerance. A pathological immune response that is abnormal and brought on by repeated exposure to the same antigen is known as hypersensitivity. Autoimmune diseases in which immune responses are directed against self-antigens, such as cancer cells, and illnesses brought on by uncontrolled or excessive responses to foreign antigens are examples of hypersensitivity diseases. These diseases are grouped into four types based on their underlying causes. They're as follows: immediate hypersensitivity, which causes an immediate response after exposure. An example of this is allergies. Antibody-mediated hypersensitivity, which causes cytotoxic reactions, meaning healthy cells die as they respond to antigens. An example of this is Graves' disease. Immune complex-mediated hypersensitivity, which occurs when antibodies bind to foreign particles, forming immune complexes that deposit in tissues and cause an inflammatory response, such as arthritis. And cell-mediated hypersensitivity, which causes due to T cells and macrophages activating and attacking the body's cells or tissues, such as type 1 diabetes. Self-tolerance failure can result in autoimmune diseases. These diseases can affect various organs and tissues and result in a variety of symptoms dependent on which cells the immune system attacks. So before we get into the rejection of transplanted organs, let me define what a transplant is. A transplant is the process of transferring cells, tissues, or organs from one location to another, usually from one person to another. This is done to correct an organ system malfunction, such as a kidney or a lung, by replacing the damaged organ with a healthy one from a donor. The immune system, as we know, has mechanisms in place to combat foreign agents, which can include transplanted organs. When one person receives an organ from another, the receiving person's immune system may recognise it as a foreign agent because the antigens on the cell of the organ differ, recognising it as non-self. Organs that are not closely matched to the recipient can result in the transplant being rejected. The likelihood of rejection can be reduced by closely matching both the organ donor and the recipient's antigens. The more similar the antigens, the less likely the rejection. Tissue typing ensures that the organ or tissue is as similar as possible, and monoclonal antibodies can be used during or after surgery to knock out the specific T cells that cause rejection. However, rejection does still happen. Hyperacute rejection occurs shortly after the transplant when the antigens are completely unmatched. For instance, if a person is given the wrong type of blood during a blood transfusion, the tissue must be removed immediately so the person doesn't die. Acute rejection usually occurs within the first few months of transplantation and is caused by the recipient's T cells recognising and attacking the foreign antigens on the transplanted organ. And lastly, chronic rejection, which can occur over an extended period of time as the body's constant immune response against a new organ gradually damages the transplanted tissue or organ. As you can see, there are many different types of autoimmune diseases which exist. Rheumatoid arthritis is a type 3 autoimmune disease in which the immune system attacks the healthy cells, sending antibodies to the linings of the joints, causing inflammation and damage. Typically, it affects the joints in the hands, wrists and knees. Although the exact cause is unknown, factors such as genes, hormones and smoking are thought to increase the risk of developing this disease. Rheumatoid arthritis affects many different systems such as joint pain and stiffness, as well as that it can cause system symptoms such as fatigue and flu-like symptoms. Multiple sclerosis, or MS, is another autoimmune disease. This is a type 4 autoimmune disease in which T cells attack the myelin sheath surrounding nerve fibres in the central nervous system, causing inflammation and nerve fibre damage. MS is a chronic disease with symptoms that vary from person to person but commonly include fatigue, difficulty walking and numbness in various parts of the body. The exact cause of MS is unknown. However, factors such as genes, a lack of vitamin D and smoking are thought to be potential causes. This final section will look at the biology of vaccination. So there are 4 categories of acquired immunity, which are as follows. Natural active immunity, which develops as a result of natural exposure to a pathogen. Artificially induced active immunity, which is gained through vaccination. The immune system is triggered by the vaccine's antigens, which prompts it to produce antibodies and memory cells targeted specifically at those antigens. Natural passive immunity, which occurs when antibodies are naturally transferred from one person to another, such as from a mother to her unborn child during pregnancy. And lastly, artificially induced passive immunity, which occurs when one person receives antibodies that have been made by someone else, such as through a transfusion. Immunological response to vaccination. A vaccination typically contains an antigen, which is a weakened or inactive form of a disease-causing pathogen. When these antigens are injected into the body, they stimulate white blood cells to produce antibodies to fight the pathogen. The World Health Organization explains it uses your body's natural defenses to build resistance to specific infections and makes your immune system stronger. Vaccinations only contain weak or harmless versions of pathogens, meaning the vaccinated person is not at risk of developing the disease. So, how exactly do vaccinations work? When a pathogen is first encountered, it takes time for the antigen to be recognised by the B lymphocytes and antibodies to be produced by plasma cells. This is known as a primary response and can take several days to develop, during which time some people experience mild symptoms such as soreness. If the pathogen is encountered again, the memory cells recognise it and quickly produce higher quantities of antibodies for a longer period, often fighting off the infection before the symptoms appear. This is a secondary response. Immunological memory to vaccination, which includes both the primary and secondary responses, is the immune system's capacity to respond to a pathogen quickly and more effectively than it did on first encounter. Based on the longevity of immunological response, different vaccinations offer varying periods of protection. For instance, the diphtheria vaccine offers protection for about 10 years, whereas the whooping cough vaccine only offers 3 to 4 years of protection. Herd immunity is the indirect protection from an infectious disease that happens when a population is immune either through vaccination or immunity developed through previous infection. Fewer people can contract the contagious disease when a large portion of the population is immunised, making it more difficult for it to spread. Those who are at risk, such as newborns, the elderly, and those unable to receive vaccinations, are also protected by herd immunity. However, before the population develops an adequate level of immunity to achieve herd immunity, several issues can arise during an outbreak. For instance, pathogens may exhibit various antigen variants, making it challenging for the body to protect against each individual antigen. Pathogens also have antigenic variability, which causes similar issues to antigen variants. Furthermore, some people do not want to vaccines either for religious or ethical reasons or due to concerns of side effects. The current state of the world's interconnectedness makes it harder than ever to achieve herd immunity, leaving us open to pandemics like the recent COVID-19 outbreak. Vaccination programs are health initiatives that aim to prevent the spread of infectious diseases. These programs are designed to reach as many people as possible, starting shortly after birth. Historically, diseases such as polio, measles, and whooping cough were common causes of death in young children. However, today, Save the Children estimate 2 to 3 million children's lives are saved every year thanks to vaccines. A recent example of a worldwide vaccination program is that of the COVID-19 coronavirus, which was first identified in late 2019, with vaccination being rolled out from December 2020. And now 69.9% of the population have received at least one dose of the vaccine. Vaccination programs are particularly important for children, with a variety of vaccinations offered from 8 weeks old, such as a 6-in-1 vaccine. Despite this, there are many factors which affect people from having vaccinations. As previously mentioned, religious and ethical factors play a significant role in some individuals' choices not to have vaccinations or not to allow their children to have them. An article published in The Lancet in 1998, which claimed the link between the MMR vaccine and autism, increased concern amongst parents regarding the safety of the vaccination. And though this link has been disproven, vaccination uptake is still only at 89%, which is 6% lower than what the World Health Organization recommends. Vaccination is one of the most effective ways to prevent the spread of infectious diseases. These diseases can't be cured with antibiotics, and the only way to control the spread is via immunisation. The Association of the British Pharmaceutical Industry state vaccination has saved more lives and prevented more serious diseases than any advance in recent medical history, and there are numerous benefits to vaccination. Vaccinations have helped to eradicate or drastically decrease the number of some diseases, for example, smallpox, which was eradicated in 1979. Vaccination also helps to reduce healthcare costs as they reduce the need for hospitalisation and treatment. Childhood mortality rates have decreased considerably. Research shows vaccination against 10 pathogens have prevented approximately 37 million child deaths between years 2000 and 2019. And as previously discussed, when a high percentage of the population is vaccinated, it creates herd immunity, which can provide protection for those who are vulnerable or cannot have the vaccination. To conclude, vaccination is a safe and effective way to prevent the spread of diseases and provides protection to the population as a whole.